This video introduces the process of cellular respiration. Before we go to the detail, I want to remind you a little bit about the microbial metabolisms and microbial cell structure. Microbial metabolisms includes anabolisms and catabolisms. Anabolisms consist of the activities which synthesize macromolecules or polymers from small molecules or monomers. Anabolisms requires energy. In contrast, catabolisms consist of the activities which break down macromolecules or polymers into small molecules or monomers. Catabolisms produce or generate energy. Cellular respiration is an example of catabolisms. Now we move to microbial cell structures. One of the significant differences between bacterial cell and eukaryotic microbial cells is that bacterial cells do not have cell organelles, and of course they also do not have a nucleus. Eukaryotic microbial cells have a typical nucleus and a number of cell organelles. The important cell organelles in cellular respiration is mitochondria. Mitochondria consists of two membranes. The space between the two membranes is called outer space, and the area at the middle of the mitochondria is called matrix. Cellular respiration includes three steps: glycolysis, Krebs or citric cycle, and oxidative phosphorylation. Because bacteria do not have a mitochondria, so the glycolysis and citric cycle takes place in the cytoplasms. Oxidative phosphorylation takes place on the cytoplasmic membrane, but for eukaryotic microorganisms, the glycolysis takes place in the cytoplasms. Krebs or citric cycle takes place in the matrix area of mitochondria. Oxidative phosphorylation takes place on the inner membrane of mitochondria. Now we will go to the detail of each of the three steps. So glycolysis can occur with or without oxygen. What exactly happens is that glucose, a 6-carbon atom molecule, is split into two pyruvate molecules. Each of pyruvate consists of 3 carbon atoms. These reactions requires ATPs, 2 molecules of ATPs, but at the end it also generates 4 molecules of ATPs. In these reactions, 2 molecules of NAD+ get reduced into 2 molecules of NADH. By the end of glycolysis, pyruvates are produced in cytoplasms, but pyruvates are not ready for Krebs or citric cycle yet. They must be converted into acetyl-CoA. The process of pyruvate being converted into acetyl-CoA is called preparatory step. Preparatory step sometimes is also included in Krebs or citric cycle, but it actually still takes place in the cytoplasms. Pyruvate has 3 carbon atoms. Acetyl-CoA has 2 carbon atoms. Therefore, when pyruvate is converted into acetyl-CoA, 1 carbon is cleaved off the molecule and is released in the form of carbon dioxide. And in this reaction, 1 molecule of NAD+ also gets reduced into NADH. Once acetyl-CoA is produced, citric cycle will take place. And again, citric cycle takes place in the matrix area of mitochondria or in the cytoplasms of bacterial cell. Krebs cycle or citric cycle requires oxygen. If after glycolysis oxygen is not available or is not around, then we will have a fermentation instead of citric or Krebs cycle. So oxaloacetic acid combines with acetyl-CoA and form citric acid. Citric acid then will undergo a number of complex reactions and will be converted back into a slow acetic acid. What's important in the citric or Krebs cycle is that NADH and FADH2 along with ATPs are synthesized or are produced. NADH and FADH2 produced from the previous steps, including the Krebs cycle, will then enter the oxidative phosphorylation process. Under optimal condition, 3 molecules of ATPs can be synthesized from 1 molecule of NADH, and 2 molecules of ATPs can be synthesized from 1 molecule of FADH2. Oxidative phosphorylation takes place on the inner membrane of mitochondria, and it also requires oxygen. So if you have to calculate the total number of ATPs from the cellular respiration, you have to keep it in mind that after glycolysis, 2 pyruvate molecules are synthesized, and each of the pyruvate molecules will enter the citric cycle. Therefore, every product after glycolysis must be multiplied with 2. Next, I will explain how NADH and FADH2 are used for ATP synthesis. Again, oxidative phosphorylation takes place on the inner membrane of mitochondria. If I zoom a portion of the inner membrane in, here is what I would say: 2 layers of phospholipids and a number of proteins. And the most important thing here is that the proteins located on the membranes of mitochondria are members of the electron transport chain, meaning they can carry electrons or electron can be passed from one molecule to another. So this is the inner membrane. One side of the membrane faces with the matrix, right? And another side faces with the outer space, the space between the 2 membranes. NADH or FADH2 will give away electrons through the process of oxidation. Electron from NADH will be passed to the first member of the electron transport chain, and then it will then be passed from it to another electron carrier. And the last electron acceptor is oxygen. The special thing about this process is that each time when electron is passed from one molecule to another, a small or a certain amount of energy is released. Because the energy levels of NADH and the members of the electron transport chain are different. So when energy is released, the energy will pump proton from the matrix area to the outer space of the mitochondria, and that keeps happening to the point that the concentration of proton of the outer space becomes very high. That makes the outer space becomes acidic. To balance out the acidity, the protons must go back into the matrix area. But because the membrane is not permeable for them, so they need to go back by the other mechanism. When the protons travel back into the matrix area from the outer space, they accidentally activate the enzymes ATP synthase. And then ATP synthase, and once ATP synthase is activated, it will convert ADPs into ATPs. So that is how ATP is synthesized from NADH.